Okay, this chapter is going to deal with linkage, recombination, and eukaryotic gene mapping. So the idea behind this chapter is that sometimes when we look at a trait or a set of traits, we find that they're inherited in in a non-Mendelian fashion, meaning that they tend to cling to each other, and the data that comes from pedigrees and from model organisms basically indicates that these traits are either the result of the same locus, or they're the result of genes that are so close together that they basically move together either during recombination or during normal segregation and assortment. Okay, and you use chi-square to determine which is which. Okay, we're looking here at an example of male pattern baldness, which can come from a lot of different reasons, including illness, injury, drugs, and heredity. The most common type seen in men is pattern baldness, technically known as androgenic alopecia, in which the hair is lost prematurely from the front and top of the head. More than 95% of hair loss in men is pattern baldness. Although pattern baldness is also seen in women, it's usually expressed weakly as a mild thinning of hair. The trait is stimulated by male sex hormones known as androgens, as shown by the observation that males who are castrated at an early age rarely become bald. Okay? SNPs from the X chromosome are not inherited independently as predicted by Mendel's principle of independent assortment. These are the results of a study trying to basically locate where the gene for male pattern baldness resided. Okay, so what they did is they went out and they found a bunch of SNPs and they located a cluster that were not inherited independently of the baldness trait. Okay, instead they tended to be inherited together, which occurs when genes are physically linked on the same chromosome and segregate together in meiosis, or they are in fact the same gene. Okay, by using mapping in families with pattern baldness, this study demonstrated that the gene for pattern baldness is closely linked to SNPs located on the short arm of the X. This region includes the androgen receptor gene encoding a protein that binds male sex hormones. Given the clear involvement of male hormones in the development of pattern baldness, the androgen receptor gene seemed a likely candidate for causing pattern baldness. More analysis showed that certain alleles of the androgen receptor gene are closely associated with the inheritance of this trait, and that variation in the androgen receptor gene is almost certainly responsible for much of the difference in pattern baldness that we see when we examine these individuals. So this is a hereditary trait. It is a sex influence trait, all right, meaning that it's expressed more prominently in men when they have the gene than it is in women, and that's due to levels of testosterone. Okay, women have less than men do, but men and women both have it. All right, and this is sex linked. It's on the X. Okay, so a linkage group is simply a group of linked genes, which are genes located close to each other on the same chromosome. So when we talk about a linkage group, we're basically talking about a cluster that's on the same molecule. Remember that if the alleles are sufficiently far apart or they're on different chromosomes, they're going to separate during meiosis, and that's Mendel's principle of segregation. Independent assortment just states that alleles at more than at one locus will sort independently from alleles at another. Okay, and again, provided that they aren't linked. Okay, recombination is just a way of rearranging the organization of particular genes on a single chromosome, and that happens at prophase 1 meiosis in both men and women. Okay. Earlier, we talked about Mendel's principles of segregation and independent assortment. So, if we want to look at these important concepts in a little more detail, segregation states that each diploid organism has two alleles at a locus, and that they separate in meiosis, and that one allele passes to each gamete. The principle of independent assortment provides additional information about the process of segregation and tells us that two alleles at a locus separate independently of alleles at other loci. The independent separation of alleles results in recombination, the sorting of alleles into new combinations. Okay? Consider a cross between individuals that are homozygous for two different pairs of alleles, A and B. Okay, so we cross a double homozygous dominant with a double homozygous recessive. The first parent, the double homozygous dominant, produces gametes with dominant alleles at both loci, and the second parent recessive at both loci. The resulting F1 are all heterozygous at both loci. Recombination means that when one of the F1 progeny reproduces, the combination of alleles in its gametes may differ from the combination in the gametes of the parents. In other words, the F1 can produce gametes with new allele combinations, such as cap A little b or little a cap b, in addition to parental gametes cap A, cap B, or little a little b. Okay? And the frequency with which that happens is proportional to the physical distance between those two genes on one chromosome. Okay? So, again, the idea here is that when this happens, right, recombinant alleles, recombinant gametes, what we've done is we have rearranged the order of genes so that we generate a novel DNA molecule, a novel chromosome that isn't present in the body cells of either parent. Okay? Crossing over results in recombination by breaking the association of genes that are close together on the same chromosome. Linkage and crossing over can be seen as processes that have opposite effects. Linkage keeps particular genes together, and crossing over is going to mix them up. It's going to scramble them. And that makes new combinations of genes. Earlier, we talked about exceptions and extensions to Mendel's principles of heredity. The concept of linkage adds a further complication to interpreting the results of crosses. However, with an understanding of how linkage affects heredity, we can analyze crosses for linked genes and successfully predict the kind of progeny we're going to make. In analyzing crosses with mixed genes, we have to know not only the genotypes of the individuals that cross, but also the arrangement of the genes on the chromosomes. So to keep track of this, we introduce a system of notation presenting crosses with linked genes. First, let's consider a cross between an individual homozygous dominant for two alleles at two linked loci and another individual homozygous recessive for these alleles at these same loci. And then we have a cross. You can see that cross here. Okay? In this notation, each horizontal line presents one of the two homologous chromosomes, and because they inherit one chromosome from each parent, the F1s will have the following genotype, as shown here. Okay? So this is our parental, this is our F1. Okay? Here, the importance of designating the alleles on each chromosome is important. One chromosome has two dominants, whereas the homologous chromosome has two recessives. The notation can be simplified with a single line instead of a double, with the understanding that the genes located on the same side of the line are on the same chromosome. So we can simplify it like this, or we can go even further and we can represent it as a slash, right? So that's on one chromosome, that's on another, right? So these three are actually equivalent ways of showing the same condition. Note that complete linkage is going to lead to non-recombinant gametes and progeny, and crossing over with linked genes leads to recombinant gametes and recombinant progeny. Okay? And the frequency of the recombinant gametes and progeny is proportional to the distance, the physical distance between the two alleles on that same chromosome, on that same DNA molecule. The apparent inconsistency between the principle of independent assortment and the chromosomal theory of heredity disappeared when biologists began finding genetic characters that didn't assort independently. One of the first cases was reported in sweet peas. Back in 1905, crosses of sweet peas which were homozygous had purple flowers and long pollen grains, with a homozygous strain that had red flowers and round pollen grains. So again, these were inbred strains, similar to what kind of Mendel did. Okay, all the F1 had purple flowers and long pollen grains, indicating that purple was dominant over red and long dominant over round. When they intercrossed the F1, the resulting F2 did not appear in the 9:3 to 3:1 ratio we expected with independent assortment. An excess of F2 plants had purple flowers and long pollen, or red flowers and round pollen. Although these researchers were unable to explain the results, we now know that the two loci that they were looking at are very close together on the same chromosome, and so they're not going to assort independently to any extensive degree. Okay, so you can see here, all right, that we've got parental phenotypes, right? Purple flower, long pollen; red flower, short pollen, right? We get the hybrid here, okay, and then we end up, all right, by having self-fertilization with a lot of flowers that are going to resemble the F1 generation, and very, very few, all right, that are the recombinant phenotype, okay, meaning that they have a collection of traits that aren't present in the parental generation, either in the F1 or the P, okay? So here you got 21 purple flower with round pollen. That doesn't exist in any of these generations. And you got 21 red flowers long pollen, which also doesn't exist in any of these generations. So these would be what we would call recombinant phenotypes, right? And these over here would be what we would call parental phenotypes, right? Where this would be the recessive condition, right? And this would be the probably the combination of the homozygous dominant and the heterozygous condition. Again, but these genes are linked. Okay, you're like getting 2 for 1 here. Okay. Genes that are close together on the same chromosome usually segregate together and are inherited together, but genes occasionally switch from one homolog to the other through crossing over. This leads to recombination and occurs between two normally linked loci. As a result, it will cause them to sort independently. Okay. The results of a test cross are diagrammed here. The heterozygote produces two types of gametes, some with the cap-M, cap-D chromosome and others with the small m, small d. Because no crossing over takes place, these gametes are the only types produced by the heterozygote. Notice that the gametes produced by the heterozygote have only combinations of alleles that were present in the parents, known as the P generation. Either the allele for the normal leaves together with the allele for tall height, or the allele for mottled leaves with dwarf height. Gametes that contain only original combinations of alleles that were present in the parents are non-recombinant or parental gametes. The homozygous parent in the test cross produces only one type of gamete, which contains small m, small d. When its gametes pair with the two types of gametes generated by the heterozygous parent, two types of progeny result. Half have normal leaves and are tall, and you can see here the other half have mottled leaves and are dwarf. Okay. These progeny display the original combination of traits present in the parental generation and are non-recombinant, or parental progeny. No new combinations of the two traits, such as normal leaves with dwarf height or mottled leaves with tall height, appear in the offspring because the genes affecting the two traits are completely linked and are inherited together. The new combinations of traits would arise only if a physical connection between these two loci were broken as a result of crossing over. So the parental or non-recombinant progeny are generally going to be the ones that are present in the largest amount if the loci are linked in the offspring. Okay. The results are different than expected when the genes assort independently. If the cap-M and cap-D loci assort independently, the heterozygous plant would produce four types of gametes: two non-recombinant gametes containing the original combinations of alleles, and two gametes containing novel combinations of alleles, such as cap-M small d and small m cap-D. Gametes with new combinations of alleles are recombinant gametes, With independent assortment, non-recombinant and recombinant gametes are produced in equal proportion. These four types of gametes join with a single type of gamete produced by the homozygous parent of the test cross to produce four kinds of progeny in equal proportion. The progeny with new combinations of traits formed from recombinant gametes are the recombinant progeny. Okay, so their progeny formed from recombinant gametes with novel combinations of traits, and the recombinant gamete is the one that possesses the novel combination of traits. So again, the percentage of recombinant progeny will increase as the physical distance between the two linked loci increases. Okay? Usually there's some crossing over between genes on the same chromosome, and that produces new trait combinations. Genes that exhibit crossing over are said to be incompletely linked. Let's see how an incomplete linkage is going to affect the result of a cross. Our theory holds that the effect of crossing over on the inheritance of two linked genes is shown here, where crossing over, taking place in prophase 1 meiosis, results in exchange of genetic material between non-sister chromatids. After a single crossover has taken place, the two chromatids that didn't participate in crossing over are unchanged, and the gametes that receive the chromatids are non-recombinant. The other two chromatids, which did participate in crossing over, now contain new combinations of alleles. Gametes that receive these chromatids are the recombinants. For each meiosis in which a single crossover takes place, two non-recombinant gametes and two recombinants are made. This result is the same that is produced by independent assortment, so if crossing over between two loci takes place in every meiosis, it's impossible to determine whether the genes are on the same chromosome or are distant or are on separate chromosomes. Okay, so that's not something you would know short of molecular analysis. However, for closely linked genes, crossing over doesn't take place in every meiosis. In linked genes where there isn't crossing over, only non-recombinant gametes are made. In meiosis in which there's a single crossover, half the gametes are recombinant and half are non-recombinant. Because each crossover leads to half recombinant gametes and half non-recombinant, the total percentage of recombinant gametes is always half the percentage of meioses in which crossing over takes place. Even if crossing over between two genes takes place in every meiosis, only 50% of the resulting gametes are going to be recombinant. Thus, the frequency of recombinant gametes is always half the frequency of crossing over, and the maximum proportion of recombinant gametes is going to end up being 50%. So to calculate recombination frequency, we take the number of recombinant progeny over the total number of progeny times 100%. One of the ideas we have to bring in at this point is coupling and repulsion of linked genes, and we can only do this if we have a dominant and a recessive version of the gene. Okay. In coupling, wild-type alleles are found on one chromosome, and the mutants are found on the other. Okay, so if they're in repulsion, all right, that's flipped. Okay, there's one mutant and one wild-type on one chromosome, and the other mutant and the wild-type on the other chromosome from the other parent. Okay, so there's cis and trans configuration, or coupling and repulsion. Okay. They basically mean the same thing. When crossing over takes place between genes for leaf type and height, two of the four gametes produced are recombinants, shown in the figure here. When there's no crossing over, all four resulting gametes are non-recombinant. Because each crossover makes half recombinant gametes and half non-recombinant, the majority of gametes made by the heterozygous parent will be non-recombinant. These gametes then unite with gametes produced by the homozygous recessive parent, which contain only recessive alleles and result in mostly non-recombinant progeny and a few recombinant. In this test cross, we see that 55 of the progeny have normal leaves and are tall, and 53 have mottled leaves and are dwarf. These are the non-recombinant, containing the original combination of traits present in the parent. Of the 123 progeny, 15 have new combinations of traits not present in the parents, and 8 with 8 having normal leaves and dwarf, and 7 having mottled leaves and tall. These are the recombinant progeny. The results of a test cross, such as the one here, reveal several things. A test cross for 2 independently assorting alleles, or 2 alleles distant on the same chromosome, is expected to produce a 1 to 1 to 1 to 1 phenotypic ratio in the progeny. The progeny of this cross clearly don't exhibit this ratio, so we might suspect that the genes aren't assorting independently. When linked genes undergo some crossing over, the result is mostly non-recombinant progeny and a few recombinant. This result is what we observe among the progeny of a test cross shown here, so we conclude that the 2 genes show evidence of linkage and some crossing over. So again, it's the same idea, right? The closer the loci are together, the fewer non-recombinant phenotypes you're going to see, because there's less distance between the 2 loci for a crossover to take place, okay? Whereas if they are distant on the same chromosome, you're not going to be able to determine the difference between the loci being on different chromosomes or being distant on the same chromosome, because if they're way far apart on the same chromosome, there will be a lot of crossing over going on. Coupling and repulsion of linked genes is just as we talked about, right? With the cis configuration, one chromosome has both wild types and one has both mutant alleles, and in repulsion, that ends up being a mutant and a wild type on each chromosome, with the mutant and the wild type for allele 1 and allele 2 being the opposite allele from the mutant and the wild type on the homologous chromosome. Okay, so we can test for this. In crosses for linked genes, the arrangement of alleles on homologous chromosomes is critical to determine the outcome of the cross. As an example, consider the inheritance of a couple of genes in the Australian blowfly. In this species, one locus determines the color of the thorax. A purple thorax, lowercase p, is recessive to a normal green thorax, lowercase p with a plus. A second locus determines the color of the puparium. A black puparium, lowercase b, is recessive to the normal brown puparium, little b with a plus. The loci for thorax color and pupation color are located close together on the same chromosome. Suppose we test cross a fly that's heterozygous at both loci with a fly that's homozygous recessive at both. Because these genes are linked, there are 2 possible arrangements of the chromosome on the heterozygous fly. The dominant allele for green thorax, small p plus, and brown puparium, small b plus, might reside on one chromosome of the homologous pair, and the recessive allele for both are going to reside on the other homologous chromosome. This arrangement, in which the wild type alleles are found on one chromosome and the mutants on the other, is the cis configuration. Another possibility is that one chromosome might carry the allele for green thorax and black puparium, and the other is going to carry the alleles for purple thorax and brown puparium. So this would be in repulsion or in the trans configuration. This arrangement, in which each chromosome has one wild type and one mutant allele, is called repulsion or trans. When the alleles in the heterozygous parent are in coupling or repulsion, that'll determine which phenotypes are most common among the progeny of the test cross. Okay? When the alleles of the heterozygous parent are in the coupling configuration, the most numerous progeny types are those with the green thorax and the brown puparium, and those with a purple thorax and a black puparium. However, when the alleles are in repulsion, the most numerous progeny types are those with a green thorax and black puparium, and those with a purple thorax and brown puparium. Notice that the genotypes of the parents in this figure are the same, okay? And that the dramatic difference in the phenotypic ratio from the progeny in the 2 crosses is entirely from the configuration of the chromosomes, be they in coupling or in repulsion. Knowledge of the arrangement of alleles on the chromosomes is important to predict the outcome of the crosses in which the genes are linked. Okay, so you can see here some examples of different situations and the progeny that come from the test cross, right? Situation of top independent assortment, right? We get the progeny assortment that we would expect if they were on different chromosomes, right? If we have complete linkage, then it's going to be 50-50, all right? And then if we have linkage with some crossing over, okay, where the genes are in coupling, more than 50% are going to be the non-recombinant and less than 50% are going to be the recombinant. Knowing the arrangement of alleles in the chromosome lets us predict the types of progeny that result from a cross that involves linked genes and allows us to determine which of these types will be the most numerous. Determining proportions of the types of offspring requires an additional piece of information, which is the frequency of recombination. The recombination frequency provides us with information about how often the alleles in the gametes appear in new combinations and allows us to predict the proportions of offspring that will result from a specific cross that has linked genes. Example here in cucumbers, smooth fruit, lowercase t, is recessive to warty fruit, uppercase T, while glossy fruit, lowercase d, is recessive to dull fruit, uppercase D. Scientists have determined that these two genes exhibit a recombination frequency of 16%. Suppose that we cross a plant that's homozygous for warty dull with a plant that's homozygous for smooth glossy and carry out a test cross on the F1. What types of proportions of the progeny would you guess? 4 types of gametes will be made. The heterozygous parent, shown here, has 2 types of non-recombinant gametes, cap T, cap D, and small t, small d, and 2 types of recombinant gametes, cap T, small d, and cap D, or cap D, little t. The recombination frequency tells us that 16% of the gametes made by the heterozygous parent are going to be recombinant. Because there are 2 types of recombinant gametes, each should arise with a frequency of about 8%. This frequency can also be represented as a probability of 0.08. All other gametes are non-recombinant, so they should arise with a frequency of 84%. Because there are 2 types of non-recombinant gametes, each should arise with equal frequency, so 42% for each. The other parent in the test cross is a homozygote recessive and therefore produces a single gamete with a frequency of 100%. 4 types of progeny result from the test cross. The expected proportion of each progeny is determined by using the multiplication rule that we talked about earlier, multiplying together the probability of each gamete. For example, test cross progeny with warty and dull fruit appear with a frequency of 42%, which is cap T, cap D from the heterozygous parent times the recessive alleles from the test cross. Okay. The proportions of the other types of F2 progeny can be calculated in a similar manner. This method can be used for predicting the outcome of any cross with linked genes for which the recombination frequencies already know. Okay. So this is kind of problem where we would give you the distance, the genetic distance, and then you could come up with the recombinants and the non-recombinants. And then we're going to complicate this a little bit further. We're going to do work with 3 genes, and you're going to determine gene order on the chromosome, right? In order to determine the difference between whether or not a set of genes are linked or not, we use the chi-square test. This test for independence lets us evaluate whether segregation of alleles at one locus is independent of segregation at another without making any assumption about the probability of the single locus genotype. In chi-square goodness of fit, the expected value is based on a theoretical relation, for example, the expected ratio in a genetic cross. For the chi-square test of independence, the expected value is based strictly on the observed values along with the assumption that they're independent. To illustrate this, we can examine the results of a cross between German cockroaches in which yellow body is recessive to brown, okay, as shown here, right? And curved wings are recessive to straight. A test cross produces the progeny shown here. If the segregation of alleles at each locus is independent, then the proportions of progeny with the y plus, lowercase y plus and lowercase y and the lowercase yy genotypes should be the same for cockroaches with the genotype of small c, small v plus, and small c, small v, and for cockroaches with the homozygous condition small c, small v. The converse is also true. The proportions of progeny with the CV plus CV and CV-CV genotype would be the same for cockroaches with genotype Y plus Y and cockroaches with genotype lowercase y recessive. To determine whether the proportions of the progeny with genotypes at the two loci are independent, we make a table of the observed numbers of progeny, like a Punnett square, except that we put the genotypes that result from the segregation of alleles at one locus on the top and the genotypes that result from segregation of alleles at the other locus on the side. Next, we compare the total for each row, the total for each column, and the grand total. These totals will be used to compute the expected values for the chi-square test of independence. Okay? So you can see the process here, step by step. Gene mapping with recombination frequencies allows us to construct a genetic map showing us the relative distance, genetic distance, of two loci on a chromosome. These are represented in what we call map units or centimorgans. And we can do this with a two-point test cross. Okay? So that is discussed here.
One of the very interesting things about genetic recombination is that you can actually use genetic recombination to figure out the distance between genes on a chromosome. And if you were to do this to all the genes on a chromosome, you could actually map out the chromosome and figure out exactly where the genes are. And we're going to explore that concept. So we're looking at a pair of homologous chromosomes, and let's just say that the orange chromosome is the paternal chromosome, so it comes from the father. And let's say that the yellow one is the maternal chromosome. And just to remind ourselves, so these are sister chromatids. That means that they are identical chromatids. They have identical genes on them. So those colored circles that I drew represent just some genes that I randomly picked. And you can see that on the two sister chromatids, I drew them in the same color because they're the same. And for that matter, these are also sister chromatids. These two yellow chromatids are also sister chromatids. And I'm just going to review a bit to give us some context. So normally in the cell, the paternal chromosome would just look something like that with the centromere middle, and the maternal chromosome would also look something like that. But during meiosis or mitosis, when they replicate, this chromosome will turn into that, so it's going to duplicate itself, and each one of those strands is a sister chromatid. And so will the maternal chromosome turn into something that looks like that. But this is still considered one chromosome, and this is considered one chromosome. And that can be a little bit confusing, because how could this be one chromosome and this also be one chromosome if this X has double the material as the original strand? And the answer to that question is because we count chromosomes by the amount of centromeres that are present. So this has one centromere, it's considered one chromosome. This X has one centromere, so it's considered one chromosome. However, they're not quite the same. This on top, it's just one chromatid, and the bottom chromosome is made up of two chromatids. So try not to get confused. Even though we're calling this one chromosome, it's really made up of two identical sister chromatids. But anyway, that was a bit of a digression. Let's focus again on our homologous chromosomes. So I picked a couple of genes that I just, put on, and we're going to focus on 3 genes in particular. And to make this a bit more real or relevant, let's just say that the green genes represent, maybe complexion, maybe like dark complexion versus a lighter complexion. And on the maternal chromosome, I also drew it in green to remind us that these are homologous chromosomes or homologous alleles. In other words, these are both alleles that code for complexion, but different shades of green because they're probably different versions. So these green genes also code for complexion. And let's say that this darker purple or magenta gene codes for, let's say, hair color. So that means that the lighter purple on the maternal chromosome also code for hair color. And then let's say that the blue genes code for eye color. So we have dark blue on the paternal chromosome and light blue on the maternal chromosome. And let's then focus on the sister chromatids where genetic recombination occurs. So these two strands are going to swap genetic information between them. Maybe like a chunk on bottom will swap, maybe something in middle will swap, maybe something on top will swap. Actually, genetic recombination also occurs between sister chromatids. However, sister chromatids are identical, so it would be of no consequence. Anyway, so let's look at the two chromatids where genetic recombination is happening, the two that I circled. Let's take a closer look at those. So here are the two chromatids that are going to exchange genetic information and undergo recombination. And I want to ask you a question. So I'm going to give you two choices, and I want you to try to figure out which one is more likely. So for the first choice, let's look at the purple and green genes. And the question is, what's more likely? Is it more likely that the purple and green genes undergo recombination? And when I say that, I mean that the genes that are originally on one chromosome separate. So I mean to say that, for example, this purple gene will get separated from that gene, and this lighter purple gene will get separated from that gene. So is it more likely that the purple and green genes recombine? Or the way I like to view it, gets separated? Or is it more likely that the blue and purple genes recombine or get separated? And the way to think about this is, well, look at the distance between them. So the distance between the purple and green genes is something like that. So in order for recombination to occur with respect to the purple and green genes, it would have to happen somewhere over here. Or on the other side, it doesn't matter, it doesn't make a difference. So you have this whole distance. Whereas if you wanted recombination to occur with respect to the purple and blue genes, you have a very small area to work with. It would have to happen somewhere here. So somewhere along, this line of the chromosome. So that means that it's much more likely for recombination to happen with respect to the purple and green genes. That is more likely because you have this entire distance to work with. If these two chromatids swap genetic information anywhere along this stretch of the chromosome, so the green and purple genes will separate or recombine. And the blue and purple genes are less likely to recombine because recombination would have to occur only in this little sliver of chromosome, and that's just smaller than the other part of the chromosome that we were looking at. So we just learned a very important concept, and that is that the further apart two genes are, the more likely it is that they will recombine. I'm actually not sure if that's the proper way to use the word, but I just use it that way. So I'm going to put it in quotes. And again, when I say recombine, I mean that two genes that were originally on the same chromosome get separated. And then the next thing we learned is that the closer two genes are to each other, I'll abbreviate each other just EO, the less likely it is that they will recombine. And now I'm going to introduce you to some terms. The centimorgan is the unit of measurement that we use to measure distance on a chromosome. And another way to say centimorgan is a genetic map unit, or M.U., which stands for map unit. And I'll give you the official definition of a centimorgan because I think it's important. It kind of ties in distance to what it has to do with recombination. And so a centimorgan is the distance between genes, just going to abbreviate between like that, for which one product of meiosis in 100 is recombinant. And to put that in simpler terms, it means that if two genes are 1 centimorgan apart, it means that 1 out of 100 times, or 1% of the time that meiosis happens, those two genes will be recombinant or separate or recombine. And we'll actually do an example of this to illustrate what this means. So here we have our two chromatids again. And let's just say that the distance between the purple and green genes is 25 map units. Remember, a map unit is the same thing as a centimorgan. And let's say that the distance between the blue and purple genes is 6 map units. So this is clearly not drawn to scale very well, but it's just an estimate. So let's first focus on the purple and green genes. So if they're 25 map units apart, so remember, if two genes are 1 map unit apart, that means that 1% of the time they'll recombine. So these are 25 map units apart. So that means that 25% of the time that meiosis happens, recombination will occur with respect to the purple and green genes. So let's see what that looks like. So we're going to see that in this spot right over here, the chromatids just swap information. So let's draw what that would look like. So let's draw our paternal chromosome, or at least part of it, and then we'll draw our, actually I'm going to draw that a little bit higher because we need more room. So here's our paternal chromosome, and then our maternal chromosome, or chromatid. And then since they kind of swapped in this spot, so I'm going to draw yellow right over here. That's the part that came from the maternal chromosome. And then I'm going to draw orange over here. That's the part from the paternal chromosome. And now let's fill in our genes. So the blue genes will just stay where they were. So we just leave them put. And the same goes for the purple genes. But then we have that piece of maternal chromosome. So we get that hunter green gene over here. And then we get the lime green gene right over here. So this is what I mean by recombination being a separation of genes. So this purple gene and that lime green gene were on the same chromosome before, but they got separated. They're not on the same chromosome anymore. And the same applies to these two genes. Now let's look at the blue and purple genes. So they're 6 map units apart. So that means that 6% of the time that meiosis happens, the purple and blue genes will get separated. So we're going to say that upwards of this spot, the chromatids swap information. So let's draw that. So here we'll draw a part of our paternal. That's the paternal chromosome and the maternal one. And they swapped somewhere like over here. So let's fill that in. So that's the part of the maternal chromosome that lands up, sorry, that's the part of the maternal chromosome that ends up on the paternal one, and the orange over here. And now let's fill in our genes. So let's first fill in the ones that just stay put. So we have that lime green gene, then we have that hunter green gene, and our purples also stay put. But the blue genes swap chromosomes. So we have our dark blue gene over here and our light blue gene over here. And again, take note of how they swap places or how they separate. So these two genes were together on the same chromosome before, but not anymore. And the same for these two genes. So let's just tie this back into the bigger concept going on. If we were to do a statistical analysis of how often certain recombinations happen, that can help us map out the genes on a chromosome.
Where we left off in the last video, we were in 1902, 1903, and Mendelian genetics had been rediscovered at the turn of the century, and Boveri and Sutton independently had proposed the chromosome theory, that the chromosomes were the location for where these inheritable factors that Mendel first talked about, where they actually were located. But we talked about in that video that that was just a theory. This was based on some observations of meiosis and seeing how chromosomes behaved, and they seemed to behave in analogous ways to some of these inheritable factors. But they really didn't have good cellular proof that chromosomes indeed were the location for these inheritable factors. And we don't really start to get that until we start looking at the work of Thomas Hunt Morgan. Now, in 1908, he decides to study fruit flies. So why does he want to study fruit flies? If you've ever seen a fruit fly, they're very, very, very small. So you could actually put a ton of fruit flies in one jar. So that's convenient. You oftentimes don't think about the practical logistics of science, but you could put a lot in one jar. They were actually cheap, and that's another practical concern of science, is you don't always have a lot of resources to do your work. And they had short lives, and they reproduced a lot. So you could very quickly get many, many offspring in many, many generations if you wanted to study how the different traits were passed on or not passed on. And so he spent some time, he started this in 1908, working with the fruit flies, and he kept breeding them in search for some type of a mutant trait. In general, when you look at traits in a species, the wild type, let me write this down, the wild type is the one that's typically seen, while the mutant trait is something that seems unusual. And after 2 years, he finally discovers a mutant trait in his fruit flies. He finds a white-eyed male. So this is the white-eyed male right over here. He says, oh, okay, now this is interesting. Let me take this white-eyed male and begin to cross it with the females. And you say, well, how does this actually occur? Well, what you do is you take a jar full of females and you put the white-eyed male in there, and then the crossing happens. And what was interesting was the inheritance pattern that he saw for this white-eyed trait. Because you have the parent generation here, but then in the F1 generation, all of the females were red-eyed, and all of the males were red-eyed. And so just off of that first generation, it wasn't clear that anything interesting was going on. But then, when he crossed these to each other, and I know what some of y'all are thinking, wait, aren't they all brothers and sisters being crossed to each other? Well, yeah, they were probably half brothers and sisters if they came from different mothers, but some of them could have been brothers and sisters. But yes, that's what people are talking about when they're crossing the F1 generation. But when they crossed these with each other, he saw a pretty interesting pattern. He saw a 3-to-1 ratio of red eyes to white eyes. So for every 4 fruit flies, he would see, let me underline these, he would see 3 red-eyed, and he would see 1 white-eyed. So the white-eyed trait makes a reappearance, which in and of itself is interesting. It shows that this can be passed on genetically. And that's interesting because this was a mutant that just showed up after he did many generations of observations. But what was even more interesting about this 3-to-1 ratio, and that 3-to-1 is something that popped up a lot in Mendelian genetics. But what was even more interesting was that he only observed the white eyes in the males in this F2 generation, in this the second generation of the crosses right over there. And so you're thinking, well, why is that a big deal? Well, he was a pretty astute guy, and he says, well, look, if I'm only seeing it in the males, and it's not like he only got 4 offspring here, it was in the ratio, he might have had hundreds of them, but it was in the ratio of 2 red-eyed females for every 1 red-eyed male for every 1 white-eyed male. And so across these hundreds in this generation, he only observed the white eyes on the males. And he said, maybe this is in some way related to the chromosome that determines sex. And so what he was able to do is say, well, let's just assume that it is. Let's assume that trait, that mutant allele, that mutant variation of the gene for eye color, let's assume it's carried on the X chromosome. And so the genotype for that first mutant fly, that white-eyed male that he found, we could call it, and this is the notation that people typically use, because this is a gene that we're assuming sits on it's sex-linked. It sits on a sex chromosome, in this case the X chromosome. The way that you would specify the genotype of that white-eyed male is, well, on his X chromosome, he had the white variation. He had the white allele, the white variation of that gene, and then on his Y chromosome, he had no variation for that gene. So we assume that it's only contained on the X chromosome. You've probably heard of heterozygous or homozygous. Well, this is a case where you're hemizygous. You only have a version of the allele on one of your two chromosomes, one of the two that you've gotten from each of your parents. So this would be the genotype right here of the white-eyed male. The genotype for the red-eyed female is specified by, so it's on the X chromosome, and the females have two X chromosomes, just like in the situation for humans. So on each of the X chromosomes, we assume that the females start off with the red allele. And the red allele, the notation is the W plus. W plus. And you might say, well, why don't we just use the letter R? Well, we could have, but the general convention in genetics is to use the letter of the first mutant type discovered for that gene, and then to use this little plus type for the wild type. So the wild type is the red eyes, and then W, which is the first mutant discovered for this gene, is the first mutant allele, that we do, so we name it after that white. So this is the white allele, and these right here, these represent the red alleles. So these are the genotype of the red-eyed female. And so when you cross that first generation, well, the white-eyed male, he can either, if he's with he'll either produce sperm that have the X chromosome in it, which is going to contain the allele, or sperm that have the Y chromosome in it, which is not going to contain the allele. And the red-eyed female, well, they produce eggs either way, either which of these X chromosomes they contribute, they're both going to have the wild type allele. And we can see how this crosses. You could get an X from both parents. If you get an X from both parents, you're going to be female, because you're going to be XX. And each of these females, since you got one wild type and one mutant type, and the wild type turns out to be dominant, they still show their phenotype is still red eyes. They still have red eyes, but now they are heterozygous. They are heterozygotes. They are carrying the white allele. Now the male offspring right over here, well, in order to be male, they got the Y chromosome from their dad. So they're not able to get that white allele, and they get the wild type from their mom. And you could see it here. And this is why all of the males in that first generation were red. They only got one copy of the allele from their wild type mother. But then what was interesting is what the crosses that you see in that next generation. If you took these red-eyed females that we already established, these are all going to be heterozygotes. And so you could see they have the red allele and they have the white allele. And you cross that with red-eyed males. You cross it with red-eyed males, what is going to happen? Well, the females in this generation, in order to be female, you have to get an X from your mom and your dad. And so they get an X from their dad, which has the wild type there, the dominant red allele. And so regardless of which one they got from their mom, they're still going to be red-eyed females. Some of them might be homozygotes, some of them might be heterozygotes. But now we see something interesting happening in the males. You could have heterozygote male flies here, where they got the X where they got the red X from their mom, or you could get the hemizygous white-eyed males where they got the white allele, the white X from their mom. And this is the exact observation that Morgan made. So it was a very interesting thing that he was able to see. He started breeding these in 1908. He started breeding these flies in 1908. It wasn't until a couple of years that he finally found that first mutant white-eyed male, and it was in 1910 and 1911 that he publishes these discoveries in Nature. And the reason why this is a big deal is he says, look, my observations are completely consistent with this I trait, this gene, being on the X chromosome. So he was able to show a direct linkage between, in this case, sex chromosomes and these heritable factors that Mendel first talked about. And he would go on, and his students that he worked with would go on to study this for many, 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 many years. And he actually ends up getting a Nobel Prize for this work. And this is a big deal because he's finally able to draw pretty substantive connections between these heritable factors of Mendel, this theory of Boveri and Sutton that maybe chromosomes have something to do with these inheritable factors. And he's showing that this is actually the case. These sex chromosomes seem to carry the trait, or in this particular case, the X sex chromosome seems to carry the gene for eye color in these fruit flies.
2 points about constructing chromosome maps from recombination frequencies need to be emphasized. First, note that we can't distinguish between genes on different chromosomes and genes located far apart on the same. If genes exhibit 50% recombination, the most that can be said about them is that they belong to different linkage groups, either on different chromosomes or on the same chromosome far apart. The second point is that a test cross for two genes that are far apart on the same chromosome tends to underestimate the true physical distance between them because the cross doesn't reveal double crossovers that might take place between two genes. When this occurs, the double crossover basically cancels out the recombination. Okay? For now, we'll just consider only double crossovers that take place between two of the four chromatids of a homologous pair. Two-strand double crossovers, which are double crossovers that take place among 3 and 4 chromatids, are going to be discussed later. Okay? A single crossover between two genes produces combinations of alleles that were not present in the original parent, and a second crossover reverses the effect, and so that restores the original combination of alleles. We therefore can't distinguish between the progeny produced by a two-strand double crossover and the progeny produced when there's no crossing over at all. As we see in sections that we're going to cover next, we can detect double crossovers if we examine a third gene that's between the two. Because double crossovers between two genes go undetected, map distances will be underestimated whenever double crossovers happen. Double crossovers frequent between genes that are far apart are a natural assumption. Okay? As a result, genetic maps based on short distances are usually more accurate than those based on long distances. And so you can see examples here of double and single crossovers and the results that take place. Okay? So let's take a look at three-point test cross.
A three-point test cross is an efficient mapping technique for finding out the order of 3 genes on a chromosome. We will use it for 3 genes, all located on chromosome 2, in Drosophila melanogaster, cherub wings, CH, black body, B, and cinnabar eyes, CN. This fly is homozygous for the mutant alleles of these genes. In contrast, a wild-type fly has longer wings, lighter body color, and slightly darker red eyes. The wild-type alleles are indicated by a superscripted plus sign. For these particular genes, the wild-type alleles are dominant to the mutant alleles. To start, homozygous wild-type flies were mated with flies that were homozygous recessive at all 3 genes. The resulting F1 progeny were therefore heterozygous at all 3 genes. In a test cross, the F1 heterozygous female flies were crossed with homozygous recessive males that had cherub wings, black body, and cinnabar eyes. The progeny in this table were produced from the test cross. Because all the flies had inherited only the mutant alleles from the male parent, these gene designations show which alleles were inherited from the female parent. For instance, out of 1,800 flies counted, 105 flies had cherub wings, inheriting mutant alleles from both parents, normal body color, inheriting a wild-type allele from the mother, and cinnabar eyes, inheriting mutant alleles from both parents. Be aware that the order of cherub, black, and cinnabar in this table is arbitrarily listed, not indicating the true order on the chromosome. Based on the information in this table, can we figure out how cherub, black, and cinnabar are positioned relative to each other on chromosome 2? First, we need to determine which of the test cross progeny are non-recombinant, also known as parental types, and which are double crossovers. The clues are hidden in the progeny counts. The non-recombinant count should be the highest, while the double crossover recombinant count would be the lowest. We determine the gene order by comparing the alleles present in the double crossovers with those present in the non-recombinants. Remember that each class of double crossovers should be like one of the non-recombinants for two loci, while differing in one locus. In this case, the cinnabar locus is different. There are three different ways cherub, black, and cinnabar can be positioned, shown here in the heterozygous genotype of the F1 female. In each, we perform a double crossover, making a crossover occur between the first and second gene, and between the second and third gene. Which one of these allele arrangements matches the classes of progeny that we identified as double crossovers? The double crossover recombinants have wild-type cherub and wild-type black together, or they have wild-type cinnabar. Of our three possible orders of loci, our third example gives the genotypes we're looking for, with wild-type cherub and wild-type black on one chromosome and wild-type cinnabar on the other. So we conclude that the correct order of the three genes is black, cinnabar, then cherub. After some practice, working from the two double crossover progeny genotypes, you will realize that a double crossover results in a middle allele that is flipped in comparison with the two end alleles. By comparing to the parental types, the cinnabar alleles have been flipped, which makes cinnabar the middle locus. These are the relative positions among the three loci on chromosome 2. Let's adjust the progeny table to reflect the correct order of the three genes. A slash between loci indicates that there has been a crossover. We can identify single crossover classes, as well as the double crossover classes and non-recombinants that we noted earlier. Now we can calculate the distance or recombination frequency between any two genes. To calculate the recombination frequency between cherub and cinnabar, we need to add the progeny resulting from a single crossover between them. And we also need to add the double crossovers, because the double crossovers include a crossover between cherub and cinnabar. We add 40, 41, 4, and 5, then divide the sum by 1800, the total number of flies counted. Multiplying by 100 gives the final answer as a percentage. The cherub-cinnabar recombination frequency, therefore, is 5%, which means cherub and cinnabar are 5 map units apart. Next, the cinnabar and black recombination frequency needs to be calculated in the same way. We add the cinnabar and black single crossover progeny counts, 105 and 102, to the double crossover progeny counts. The sum is divided by 1800 and multiplied by 100, resulting in 12%, or 12 map units. With these two frequencies calculated, we can place these loci in their relative positions to one another on a chromosome map. Cherub and cinnabar are 5 map units apart, while cinnabar and black are 12 map units apart.
While genetic maps can be constructed from a series of test crosses for pairs of genes, this approach isn't particularly efficient because numerous two-point crosses have to be carried out in order to establish the order of genes and because double crossovers are going to be missed. A more efficient mapping technique is a test cross for 3 genes, or a 3-point test cross. With a 3-point test cross, the order of the 3 genes can be established in a single set of progeny, and some double crossovers can usually be detected, providing more accurate distances on the map. So in a 3-point test cross, we have a cross between an individual heterozygous at 3 loci and an individual homozygous recessive at those same loci. So let's think about what happens when crossing over takes place in 3 hypothetically linked genes. The figure here shows a pair of homologs of an individual that's heterozygous at 3 loci, A, B, and C. Notice that the genes are in coupling configuration. All the dominants are on one chromosome and all the recessives are on the other. 3 kinds of crossover events could take place between these 3 genes. two types of single crossovers, and a double crossover. In each type of crossover, two of the resulting chromosomes are recombinant and two are non-recombinant. Notice that in the recombinant chromosomes resulting from the double crossover, the outer two alleles stay the same as in the non-recombinant, but the middle allele is different. This result provides us with an important clue about the order of the genes. In progeny that result from a double crossover, only the middle allele should differ from the alleles present in the non-recombinant progeny. So you can see that illustrated here. Okay. When we talk about genetic maps with a three-point test cross, this allows us to determine gene order and determine the location of crossovers. So the idea here with gene order is expressed in this tutorial.
We will use Drosophila to see how a three-point test cross can reveal the order of mutant loci on a fly chromosome. In the triple mutant fly, scarlet eyes are recessive to wild-type red eyes, ebony body color is recessive to gray body color, and spineless bristles are recessive to normal bristles. All three mutations are linked on Drosophila chromosome 3, but in what order? We set up a three-point test cross to examine recombination among the mutant loci. Our triple mutant male parent is homozygous for all three mutations. All male gametes will carry all three recessive mutations. No recombination can occur. Our wild-type female parent is heterozygous for all three mutations, and the mutations are coupled on one chromosome. Meiotic recombination in this parent should be easy to detect in her progeny. Let's take a look at crossing over during meiotic recombination. Recall that in a three-point cross, two crossovers are needed to produce a double recombinant during meiosis, one on each side of the middle gene locus. As a result, double recombinants are the least common meiotic products. Non-recombinants with no crossovers are the most common products. The middle gene locus is always the one which is recombined by such double crossovers, as we see here in the gametes, and eventually in the double recombinant progeny. By doing the three-point fly cross, we can easily learn which mutation maps between the other two. Our test cross produces 755 progeny, expressing 8 different phenotypes. As expected, the most common progeny are non-recombinants, wild-type heterozygotes, and triple mutant homozygotes. The double recombinants, spineless flies, and scarlet-eyed ebony flies are easy to identify. They're the two least common classes of progeny flies. Spineless progeny differ from wild-type non-recombinant progeny at a single locus, spineless. Similarly, scarlet-eyed ebony progeny differ from the triple mutant non-recombinants at a single locus, again spineless. Clearly, spineless is the double recombinant locus that maps between the other two mutations.
To examine gene mapping with a three-point test cross, we can look at 3 recessive mutants in Drosophila. In this species, scarlet eyes are recessive to wild-type red, while ebony is recessive to wild-type gray body color, and spineless, which is the presence of bristles, is recessive to wild-type normal bristles. The loci encoding these characters are linked and located on chromosome 3 in the fruit fly. We'll refer to these 3 loci as ST, E, and SS, all lowercase. But keep in mind that either the recessive allele or the dominant allele can be present at each locus. So when we say that they are 10 map units between ST and SS, we mean that there are 10 map units between the loci at which the mutations ST and SS occur. We could just as easily say that there are 10 map units between ST plus and SS plus. To map these genes, we need to determine their order on the chromosome and the genetic distance between them. First, we have to set up a three-point test cross where we take a fly heterozygous at all 3 loci and a fly homozygous for recessive alleles at all 3 loci and cross them. This generates an F1 heterozygote for all 3 loci. We might cross a stock of flies that are homozygous for the wild-type allele at all 3 loci with flies that are homozygous recessive at all three loci and generate F1 hets. Okay? The order of the genes has been arbitrarily assigned because at this point we don't know which one is the middle gene. Additionally, the alleles in the F1 hets are in coupling configuration because all the wild-type dominants are inherited from one parent and all the recessives from the other, although the test cross can also be done with the alleles in repulsion. In the 3-point test cross, we cross the F1 hets with flies that are homozygous for all three recessive mutants. In a lot of organisms, it doesn't make any difference whether the het parents in the test cross are male or female, provided that the genes are autosomal. In Drosophila, no crossing over takes place in males. It's just a peculiarity of the species. Because crossing over in the het is essential for determining recombination frequency, the het flies in our test cross must be female. So we mate female F1s that are hets for all three traits with male flies that are homozygous for the recessives. The progeny of the cross are listed in the figure. For each locus, 2 classes of progeny are made. Progeny that are hets, displaying the dominant trait, and progeny that are homozygous, displaying the recessive. With 2 classes of progeny possible for each of the 3 loci, there will be 8 classes of phenotypes possible in the progeny. To map these genes, we need information about where and how often the crossing over takes place. In the homozygous recessive parent, the 2 alleles at each locus are the same, so crossing over won't have any effect. We won't see it. Okay? With or without crossing over, the gametes from this parent have 3 recessive alleles. However, in the het parent, we have different alleles on the 2 chromosomes, and crossing over can be detected. The information that we need for mapping as a result comes entirely from the gametes in the female fly, because chromosomal contributions from the homozygous parent are not going to give us any information. Whatever alleles are present on the chromosome contributed by the heterozygous parent are going to be expressed in the progeny. Okay? So the steps to determine gene order is to identify the non-recombinant progeny, identify the double crossover progeny, compare the phenotypes of the double crossover with the phenotypes of the non-recombinant progeny. They should be alike in 2 characters and different in 1. The characteristic that differs between the double crossover and the non-recombinant progeny is the middle gene. When we write the results of a 3-point test cross with the loci in the correct order, we get the locations of the different genes. These results from the test cross are illustrated here with the loci shown in the correct order. The location of a crossover is indicated by a slash. The sex of the progeny has also been designated arbitrarily. Okay. Drosophila has 4 linkage groups corresponding to its 4 pairs of chromosomes. These genes were mapped using recombination frequencies, and the distances between them within a linkage group are map units. Note that small chromosome 4 never undergoes recombination. There's just not enough distance there for that to take place. When we calculate recombination frequencies, we take the sum of all single and double crossovers over the total number of progeny. Interference and coefficient of coincidence basically express the number of observed double crossovers over the number of expected, while the interference is 1n minus the coefficient of coincidence. Okay? The effect of multiple crossovers in mapping human genes will be addressed next. Note that so far we've examined the effects of double crossovers taking place between only 2 of the 4 chromatids of a homologous pair. Double crossovers that include 3 and even 4 of the chromatids of a homologous pair can take place as well. As we've seen, if we examine only the alleles at loci on either side of both crossover events, 2-strand double crossovers result in no new combinations of alleles and no recombinant gametes are made. 3-strand double crossovers result in 2 of the 4 gametes being recombinant, and 4-strand double crossovers result in all 4 gametes being recombinant. Thus, 2-strand double crossovers produce 0% recombination, 3-strand double crossovers produce 50%, and 4-strand produce 100%. The overall result is that all types of double crossovers taken together produce an average of 50% recombinant gametes. As we've also seen, two-strand double crossovers cause alleles on either side of the crossover to remain the same and produce no recombinant progeny. 3- and 4-strand double crossovers produce recombinant progeny, but these progeny are the same types as those made by single crossovers. As a result, some multiple crossovers go undetected when the progeny of genetic cross is observed. Therefore, map distances based on recombination rates underestimate the physical distance between the genes because some multiple crossovers aren't detected in the progeny. When genes are very close together, multiple crossovers are unlikely, and genetic distance based on recombination rates accurately corresponds to physical distance on the chromosome. But as the distance between the genes increases, more multiple crossovers are likely, and the discrepancy between genetic distance and physical distance is going to go up. To correct for this discrepancy, scientists have developed mathematical mapping functions which relate recombination frequency to actual physical distance, as shown here. Most of these functions are based on the Poisson distribution, which predicts the probability of multiple rare events. With the use of such mapping functions, map distances based on recombination rates are more accurately estimated. Okay? So the idea here basically is that when you're just looking at phenotypes of progeny, you're going to have some crossover events that aren't going to distinguish in phenotype because they'll cancel each other out, right? Whereas odd crossover numbers between two loci are going to be detected as shuffled phenotypes in the progeny when you do your test cross. Okay. We also need to throw in the fact that it's been reported that there are chromosomal regions called crossover hotspots that are occasionally found between loci. So a higher than normal number of crossover events can happen even in loci that are close together in certain circumstances. Okay, so that's another complicated factor. Efforts to map human genes are hampered by the inability to perform specific crosses and the small number of progeny in most families. Scientists are often restricted to the analysis of pedigree, which can be incomplete and provide limited info. Nevertheless, a large number of human traits have been successfully mapped with the use of pedigree data to analyze linkage. Because the number of progeny from any one mating is small, data from several families and pedigrees is combined to test for independent assortment. The methods used in these types of analyses are complex, but an example will illustrate how we can do this. One of the first documented demonstrations of linkage in humans has been for the locus for Nail-Patella syndrome and the ABO blood type. Nail-Patella syndrome is an autosomal dominant disorder characterized by abnormal fingernails and no kneecaps. The ABO blood types are determined by autosomal loci and multiple alleles. Right? We talked about that. That's an example of multiple alleles and codominance and dominance. Right? Linkage between the genes encoding these two traits was established in families in which both traits were segregated. A pedigree for part of one such family is shown here. Nail-Patella is so rare, we can assume that people who have this trait are heterozygous for it. Unaffected people are homozygous. The ABO genotypes of the individuals can be inferred from their phenotype and the types of offspring that they make. Female 1-2 in this figure has blood type B, which has two possible genotypes. Cap I big B, Cap I big B, or Cap I big B little I. Okay? So BB or BO, basically. Right? Because some of her offspring are blood type O, she must have therefore inherited a small I allele from each parent. Female 1-2 must have genotype Cap IB little I. Similarly, the presence of blood type O offspring in generation 2 indicates that male 1-1 with blood type A has to also carry an I allele and therefore has a genotype Cap I, Cap A, little I. Okay, or we could say AO. I want to make it easy. The parents of this family are shown here. Okay, up top. Right. In generation 2, we can see that the genes for the Neil-Patella syndrome and the blood types don't appear to assort independently. All children in generation 2 with Neil-Patella have either blood type B or O, and all those with blood type A have normal nails and kneecaps. If the genes encoding Neil-Patella syndrome and ABO blood type assorted independently, we'd expect that some of the children in generation 2 would have blood type A and Neil-Patella, inheriting both cap A, and N alleles from their father. The observed outcome indicates that the arrangement of the alleles on the chromosome of the crossed parents are as shown here. Okay? The pedigree indicates that there is no recombination among the offspring of the parents, but there are 2 instances of recombination among the offspring in generation 3. Individuals 2-1 and 2-2 have the following genotypes. Okay, so you can see here 2-1 and 2-2, right? There's 2-1, right? And there's 2-2. Okay? So they must have inherited both this little I and the little n from the father. These alleles are on different chromosomes in the father, so crossing over must have taken place. Crossing over must also have taken place to produce child 3-3. So you can see here, child 3-3. Okay, crossover event. Okay. In this pedigree, 13 children are offspring of matings in which the genes encoding Neil-Patella and ABO segregate. 2 of the 13 children are recombinant progeny, and on this basis we might assume that the loci for Neil-Patella syndrome and ABO are linked with a recombination frequency of 2 in 13, which is 15.4%. However, it's possible that the genes are assorting independently and that the small number of children just makes it seem as though the genes are linked. To determine the probability that genes are actually linked, we often calculate the load score. To obtain a load score, we calculate the probability of obtaining the observed result under the assumption that the genes are linked with a specified degree of recombination, and the probability of obtaining the observed results under the assumption of independent assortment. We then determine the ratio of the 2 probabilities, and the logarithm of the ratio is the load score. As an example, suppose that the probability of obtaining a particular set of observation under the assumption of linkage and a certain recombination frequency is 10%, and the probability of obtaining the same observation under the assumption of independent assortment is basically 1 in 10,000. All right. The ratio of these 2 probabilities is 0.1 over 0.0001, which is 1,000, the logarithm of which is 3. Thus, the linkage with the specified recombination frequency is 1,000 times as likely as independent assortment to produce what was observed. A load score of 3 or higher usually is considered convincing evidence for linkage. So this is basically another way of getting the same kind of information that a chi-square might give you. Okay? The load score of the ratio is the ratio of the log of the ratio of the probability of obtaining a set of observations, assuming a specified degree of linkage, to the probability of obtaining the same set of observation with independent assortment, and is used to assess the likelihood that linkage is taking place here. Okay? So that's load score. Other mapping techniques include genome-wide association studies, which look at non-random associations between the presence of a trait and alleles at different loci scattered over the genome, which basically is looking for association between traits and particular suites of alleles in a population. Linkage disequilibrium is defined as non-random association between genetic variants within a haplotype, which is defined as a specific set of linked genetic variants or alleles on a single chromosome or part of a chromosome. Okay? Imagine that we're interested in finding genes that contribute to bipolar disorder, okay, which is a disorder in which you have cycling moods between severe depression and mania. Right? When a mutation that predisposes a person to bipolar arises in the population, it will occur on a particular chromosome and be associated with a set of alleles on that chromosome. In the example shown here, The D minus mutation first comes on a chromosome that has alleles A and B2 and C4, and therefore the D minus mutation is initially linked to A2, B2, and C4. A specific set of linked alleles, such as this, is called a haplotype, and the non-random association between alleles in the haplotype is linkage disequilibrium. Because of the physical change between the bipolar mutation and the other alleles of the haplotype, bipolar and the haplotype will be inherited together. Crossing over, however, breaks the association between the alleles in the haplotype and reduces the linkage disequilibrium between them. How long the linkage disequilibrium persists over evolutionary time is going to depend on the frequency of recombination between alleles at different loci. When the loci are far apart, the disequilibrium breaks down quickly. When the loci are close together, crossing over is less common and linkage disequilibrium persists longer. The important point here is that linkage disequilibrium provides information about the distance between genes. A strong association between a trait such as bipolar disorder and a set of linked genetic markers indicates that one or more genes contributing to bipolar disorder are likely to be near the genetic markers. Other techniques that we can use to determine distance between genes include somatic cell hybridization, deletion mapping, and physical mapping using molecular analysis, where we actually look at the chromosomes physically. Okay? So, in somatic cell hybridization, we have the fusion of different types of cells. Most mature somatic cells can undergo only a limited number of divisions and can't be grown continuously, but cells that have been altered by virus or derived from tumors have lost those constraints and will divide indefinitely. This type of cell can be cultured in the lab and produce a cell line, which is basically a artificially grown tissue that contains genetically identical cells that divide indefinitely and can be cultured in what we call tissue culture dish. Okay? Fusion of somatic cells is basically somatic cell hybridization. Okay? So there are chemical agents that can afford us to do that. Cells from two different cell lines can be fused by treating them with polyethylene glycol, also called PEG, or other agents that alter their plasma membrane properties. The resulting cell has two nuclei and is called a heterokaryon. The two nuclei of a heterokaryon eventually also fuse and generate a hybrid cell that contains chromosomes from both cell lines. If human and mouse cells are mixed in the presence of PEG, their fusion results in human-mouse hybrids. The hybrid cells tend to lose chromosomes as they divide, and for reasons that aren't understood, chromosomes from one species are lost preferentially. In human-mouse somatic hybrids, the human chromosome tends to be lost where the mouse chromosomes get retained, and eventually the chromosome number stabilizes when all but a few human chromosomes have been lost. Chromosome loss is random and differs in cell lines, and the presence of these extra human chromosomes in the mouse genome makes it possible to assign human genes to specific chromosomes. The heterokaryon, thus, is the cell that has two nuclei derived from different cells through cell fusion. So basically, you fuse the cells, you let them grow and divide, and eventually you get down to one or two human chromosomes in that tissue culture. Okay, and then you can look for a gene product. Okay, in that cell culture. Okay. When we do somatic cell hybridization, we have a panel of different hybrid lines. Each line is examined microscopically, and the human chromosome that it contains is ID'd. The cell lines of the panel are chosen so that they differ in the human chromosome that they have retained. As an example, one hybrid line might possess human chromosomes 2, 4, 7, and 8. Another might have chromosomes 4, 19, and 20. Each cell line in the panel is examined for evidence of a particular human gene. The human gene can be detected by looking either for the gene itself, such as using PCR, for the protein that it makes, using things like antibodies. Correlation of the presence of the gene with the presence of the specific human chromosome allows the gene to be assigned to the correct chromosome. As an example, if a gene is detected in both of the aforementioned cell lines, the gene must be on chromosome 4 because it's the only human chromosome common to both lines. Somatic cell hybridization is used less often today because DNA sequencing and mapping with molecular markers can often establish the chromosomal locations of linkage groups. Another method of determining the location of a gene is deletion mapping. Special staining methods have been developed that reveal characteristic banding patterns on chromosomes. The absence of one or more of these bands are normally seen on a chromosome and reveal the presence of a deletion, which is a loss of a segment of a chromosome. We can assign genes to regions of chromosomes by studying the association between a phenotype or a product encoded by a gene and a particular deletion. So if we knock out a particular region of a human chromosome, say in a heterokaryon, right, or in another type of cell line, and then we probe for a putative gene product in that deletion clone, and we don't see it, then we can assume that it was made by genetic material that was within the deletion. Okay? So these are methods that determine the location of a gene on a chromosome indirectly. We now can look directly at chromosomes. We're able to use a method known as FISH. Fluorescence in situ hybridization determines the chromosomal location of a particular gene by molecular analysis. This requires the creation of a probe, which is a single-stranded DNA complement to the gene of interest. The probe will shine when a laser hits it and glow a particular color reflected by the dye attached to the probe. Okay, we can visualize this with a fluorescence microscope. Okay, the presence of the fluorescence from the bound probe reveals the location of the gene on a particular chromosome. So this is the most direct way outside of sequencing that we know how to locate a gene. In addition to allowing us to see where a gene is located on a chromosome, modern lab techniques let researchers identify the precise location of a gene at the nucleotide level. As an example, DNA sequencing gives us physical distance between loci. And that's the ultimate, right? You just sequence the whole damn thing, and you know the physical distance between the loci of interest. In recent years, we've studied variation in rates of recombination and found that they vary. They vary among species and among and along chromosomes of single species, and even between males and females of the same species. As an example, about twice as much recombination takes place in humans as in mice and rats. Within the human genome, recombination rates vary among chromosomes, with chromosomes 21 and 22 having high recombination rates and chromosomes 2 and 4 having low rates. We've also detected differences between male and female humans in rates of recombination. Autosomal chromosomes of females undergo about 50% more recombination than autosomal chromosomes of males. Geneticists have found numerous recombination hotspots, where recombination is 10 times higher than the average elsewhere in the genome. The human genome contains an estimated 25,000 to 50,000 such hotspots, and about 60% of all crossovers take place here. For humans, recombination hotspots tend to be found near but not within the active gene. Recombination hotspots have been detected in genomes of other organisms as well. Other chromosomal regions, like those near centromeres, often display reduced rates of recombination. And that may have to do with the conformation of the DNA in the centromere, okay? It being more densely packed and less likely to participate in a recombination event because it's more difficult to unwind, okay? Just as one example. Okay, I thank you for listening, and I will see everybody in the next podcast.